Hi, I'm Clara. I'm a PhD candidate in computer science at UC Berkeley, and my talk today is titled Prediction-Powered Inference. And first things first, I'd really like to thank the organizers for inviting me to share this work, and especially for accommodating a recorded talk. And so in this talk abstract, I described two stories, but at the end of an exciting full-day session, I thought it'd be more fun and engaging to focus on one story titled Prediction-Powered Inference. So the question behind this talk is, how can we use machine learning predictions to make trustworthy scientific conclusions? So we, as a scientific community, are increasingly turning to machine learning systems as high throughput, inexpensive proxies for what would have been expensive, low throughput gold standard experiments. These systems include AlphaFold and other protein structure predictors, predictive systems for phenotypes, gene expression, predictive models for potential viral hosts, as we've seen in talks in this session, and even the outputs of protein language models and other large language models trained on biological sequences. And we're all very excited about how to use these predictions to accelerate the process of science. However, there's a problem, and that's that predictions contain errors. We're all aware of this. So when we draw scientific conclusions using predictions, how do we know we can trust those conclusions? To address this, we propose a new framework called prediction-powered inference. This work was done with these wonderful collaborators here. And I'm going to start this with a motivating story about post-translational modifications. So post-translational modifications, or PTMs, are modifications of specific sites of a protein sequence that play a host of regulatory roles throughout the proteome. And in 2004, researchers made an observation in this paper that the sequence regions where we tend to find PTMs look very similar to sequence regions that are intrinsically disordered or that don't have a fixed 3D structure. So this was an insightful observation that would go on to motivate many lines of research. But back in 2004, it turns out they couldn't actually quantify or estimate the association between disorder and PTMs. And the reason is because there simply weren't enough high-quality experimental protein structures. Now, fast forward almost 20 years, and we have AlphaFold for predicted structures for hundreds of millions of natural protein sequences. And last year, a very exciting study came out that used these abundant predictions to finally quantify this association that had been hinted at in 2004. And they did this by computing this odds ratio. So this is the probability of being in an intrinsically disordered region, given that there is a post-translational modification, or PTM, divided by the probability of being in an IDR, given that there's no PTM. So this odds ratio is just a standard way to quantify association between two binary random variables. You can think of it like a Pearson correlation coefficient, but just for binary values. So if the value of this is greater than 1, that tells you these things are positively correlated. And if it's less than 1, they're negatively correlated. All right. The twist here is that this study wasn't actually looking at IDRs. They were looking at predicted IDRs based off of the AlphaFold predicted structure. So this story points to a bigger story happening all over science, which is that predictions are being used for scientific inquiry. And we're seeing this not just in proteomics, but in studying regulatory DNA, disease-causing variants, the demographics of patient phenotypes, all over microbiology, as you've seen in the session today, and even beyond biology in studying human welfare more broadly. And generally, it makes sense to be doing this. We all know gold standard experiments can be incredibly expensive, time-consuming, low throughput, resource intensive, whereas predictions are super cheap, they're basically instantaneous, and they're very easy to acquire. Now, despite these advantages, it should make you a little uncomfortable or concerned when predictions are used to draw scientific conclusions. And the reason is we all know they're simply not the same quality as gold standard experiments. So if I draw a scientific conclusion based on looking at a bunch of AlphaFold predicted structures, if I don't do experimental validation, how do I know that those conclusions wouldn't have been slightly different if I'd been looking at high-quality experimental structures instead? So prediction-powered inference is a new statistical framework we propose for doing provably valid statistical inference when using predictions as data. So here's the problem setting that we address. We have some labeled data consisting of features X and a label Y. So Y is anything you would typically measure with a gold standard experiment. So for example, X can be protein sequences and the Ys can be their structures, or X's can be promoter sequences or other regulatory elements. Y can be the corresponding gene expression levels, etc. And I'm sometimes going to call this labeled data the gold standard data as well. So in addition to our gold standard data, we'll also have access to a much, much larger set of unlabeled data, indicated by these super wide bars. So in this unlabeled data, we also have features X prime, but here the labels Y prime are unobserved. We didn't do gold standard experiments for them. So you can think of this as, for example, all possible protein sequences I could be interested in, but which I don't have experimental protein structures for, or all possible metagenomic sequences or regulatory elements that I don't have phenotypes or species labels for, etc. Importantly, for both the labeled data and the unlabeled data, I can get predictions, F and F prime. And these are made using any machine learning model you think is appropriate that's already been trained on separate data. We're going to focus on a common setup in science, which is that there's an estimand that we want to know the value of. And this estimand, called theta star, is going to be some property of the underlying data distribution. So for example, it could be the mean value of Y, it could be the some quantile of Y, it could be some regression coefficient between Y and X, etc. So to make that just a bit more concrete, the mean value of Y can often be the frequency of some label of interest. So it could be the frequency of a certain phenotype in a population, the frequency of some protein structural feature, the count of a certain label, like the count of potential hosts or infections in a geographic region, etc. And of course, the regression coefficients will be relevant whenever you're interested in the association between the label Y and some feature in X. Now, our goal is going to be to use all this data to construct a confidence interval that contains the true value of the estimand with guaranteed high probability. And we're going to call this property validity. So our goal is to construct confidence intervals that are valid. So how might you do this with existing tools today? In what we'll call the classical approach, you could just use the labeled gold standard data and totally ignore the predictions. So this is basically how science has been done for almost a century since the birth of modern statistics. You just look at your gold standard experimental data, and you use existing statistical tools which leverage, for example, central limit theorem to construct confidence intervals that are valid. They'll have this guarantee. So that's great. That's how science has been done. It works. It produces valid intervals, valid statistical inference. But the downside of the classical approach is that you're completely losing out on the information contained in predictions. So even though predictions have error, we all know that they are still quite informative. That's why we're so excited about systems like AlphaFold. They can produce extremely accurate predictions, even though there's sometimes a little bit of error. So if you're just totally ignoring the predictions, you are basically tossing out evidence that you could have been incorporating into your scientific inquiry. So on the other extreme, in what we'll call the imputed approach, you could imagine just looking at the predictions and treating it as gold standard labels. And if you do that and use existing statistical tools to construct confidence intervals, those intervals will be invalid. They're not actually going to contain the ground truth value because of prediction error. So prediction-powered inference is a framework for getting the best of both worlds by using both the labeled gold standard data and the predictions on the unlabeled data. So prediction-powered inference will be able to give you confidence intervals that are provably valid while also squeezing information from the predictions to effectively increase sample size. So before we see exactly how this works, I'm going to show a quick demonstration using data from that Blue Dow et al. Paper that used AlphaFold predicted structures to compute this odds ratio on the x-axis. So for this demo, we looked at the portion of their data that did have gold standard intrinsic disorder labels. And on that portion of the data, we can compute the true odds ratio, which is this dotted vertical line. We then randomly threw out the vast majority of those labels, pretend we never saw them, And instead, we got disorder predictions using AlphaFold predicted structures. In this simulated setup, we then used the classical approach, imputed approach, and our approach, prediction-powered inference, to construct confidence intervals for this odds ratio. All right, so before I show those, remember that we want two things from a confidence interval. First and foremost, we want it to be valid, meaning it has to contain the true odds ratio, right? We want a procedure that's leading us to some scientific truth. Secondly, we would also like these intervals to be small so that they're actually informative. All right, so here's the classical interval in gray. As expected, it's valid. It contains the true value, but it's also very, very wide. And that's because there's just a very small amount of labeled gold standard data. On the other hand, this imputed interval in orange is extremely small due to the immense number of AlphaFold predicted structures available, but it's invalid. It totally misses out on the true odds ratio. So the prediction powered interval in green gives us the best of both worlds. It's valid, contains the ground truth value of the odds ratio by correcting for prediction error, but it also squeezes information from the predictions to effectively increase sample size. And that means that the intervals are much smaller and more informative. So this is a high-level pictorial summary of what prediction-powered inference can offer you. It allows you to take advantage of the power and scale of your predictive systems to do science while still doing provably valid statistical inference. Okay, so how exactly does this work? On this slide, I'm just going to sketch out the high-level principle and intuition behind prediction-powered inference. If you're interested in the more concrete technical details about how the mathematical mechanism works, those details are all in our preprint, so please feel free to look at that. So prediction-powered inference starts with identifying what we call the rectifier. And the rectifier is a specific characterization of prediction error that is relevant for your estimate of interest. So if you were interested in estimating the mean value of y versus estimating some regression coefficient between y and x, you can imagine that prediction error is going to affect those two goals differently. So the rectifier is the particular notion of error that is relevant for your estimate that you care about. And in our work, we provide a general recipe for mathematically identifying and constructing the rectifier. So just to map this onto this diagram, in this diagram, the orange point on the right is theta star. That's the true value that we want to estimate. The orange point on the left is theta tilde f. That's the estimate you would get solely based on the predictions. And that's typically going to be biased because of prediction error. And in this diagram, delta f is the rectifier. And that basically represents, again, that notion of error that you need to correct your prediction-based estimate and get it to the true value. So for just a bit more intuition, if this helps, if you were trying to estimate the mean value of y, the rectifier in this case is actually just the bias. It's the expected difference between the predictions f and the gold standard labels y. So that's the example of the rectifier for the mean value of y. And again, we provide a general recipe for mathematically constructing the rectifier for a broad class of other estimates that you might care about. Okay, so the second step is to construct a confidence interval on that rectifier. So the rectifier is a mathematical quantity that needs to be estimated. So we use the labeled data to build a confidence interval and estimate that rectifier. And that confidence interval is called R. Finally, we're going to form our prediction-powered confidence interval by propagating theta tilde f, that estimate based on the predictions, through all possible values of the rectifier in the confidence interval R. So this prediction-powered confidence interval in green basically contains all values of theta that result from taking the prediction-based estimate and correcting it with a value of the rectifier. And so that's the high-level intuition between the mathematical procedure that we're proposing. I'm also going to note that what we propose has this nice intuitive property that the better your predictions are, so the closer f is to y, then the smaller the rectifier will be. And the smaller the rectifier is, the smaller your resulting confidence intervals. So that kind of matches our intuition that the better your predictions are, the more powerful your scientific inferences can be, the smaller the intervals that you get out at the other end. Okay, so I'm just going to make this a bit more concrete and briefly overview our main technical result, which is just that the procedure we proposed, prediction-powered inference, gives provably valid confidence intervals for a broad class of estimands that we characterize mathematically. So some examples of estimands that we can provably handle include, again, the mean value of y, the median or other quantiles of y, and any sort of generalized linear regression coefficient between y and x. So this includes linear, logistic, or Poisson regression coefficients, also lasso regression coefficients. And again, to instantiate this more concretely, if you're interested in the frequencies of various labels such as phenotypes or structural features, or if you're interested in correlation coefficients or regression coefficients between labels and different features, those would all be handled by the estimands that we can cover. So this is a brief version of our main theorem. The details aren't super important. The main point is just that we produce confidence intervals that are provably valid. They're proven to contain the true value of the estimand theta star with guaranteed high probability. All right, so beyond the theory, I also want to emphasize that the algorithms for implementing prediction-powered inference are incredibly practical and easy to use. So as an example, this is the implementation for the prediction-powered confidence interval for the mean value of y. And you can see that it's just 10 lines of Python code. And it's quite straightforward. It's basically just estimating the rectifier and correcting the prediction-based estimate with that rectifier. Here's another example. This is an implementation of the prediction-powered confidence interval for a quantile of y. So this one's a little bit more involved because the rectifier for the quantile is a little bit more sophisticated mathematically. But here, still, it's only about 24 lines of Python code. So the algorithms for doing prediction-powered inference are very practical, very simple, super applicable to a broad class of estimands that you might be concerned with for your particular application. All right, so I'm going to finish by demonstrating our framework in action on a few different data sets. I'm first going to revisit that Budao et al. Paper that I was inspired by for this work. So here, I'm performing the same experiments I described earlier, where we're constructing confidence intervals for this odds ratio between disorder and post-translational modifications. And here we're doing it for two different types of post-translational modifications. There's phosphorylation on the left and ubiquitination on the right. And in both cases, you can see that the imputed intervals in orange are these extremely small orange intervals that are pointed at with the orange arrows. And they're so small because of the immense number of predictions available through AlphaFold predicted structures. However, they're invalid. They don't contain the true odds ratio value. In contrast, our prediction-powered intervals account for prediction error and are provably valid. So these green intervals contain that true value. Simultaneously, they're much smaller than the classical intervals, which totally ignore predictions and only use gold standard data. And we see that trend of the prediction-powered intervals being smaller than the classical ones across a wide range of values of the labeled data. So on the x-axis in these graphs, little n refers to the number of labeled data points. And across the whole range, we see that the widths of the prediction-powered intervals in green is smaller than the classical ones. And again, this basically means that our prediction-powered intervals are effectively increasing sample size by squeezing information from the predictions. So there's a whole number of super exciting applications we walk through in the paper. I'm just going to highlight two more here. So here we're doing gene expression analysis with a predictive model of gene expression that's a transformer. So here we have a small amount of labeled data consisting of features, which are promoter sequences, and labels, which are experimentally measured gene expression caused by those promoters. In addition, we have an extremely large unlabeled data set of native yeast promoter sequences, and we have predictions for gene expression given by a state-of-the-art transformer model for predicting gene expression. And the goal here was to estimate or put a confidence interval on different quantiles of gene expression induced by this population of native yeast promoters. So on the left, we're looking at the 0.5 quantile or median gene expression. And on the right, we're looking at the 0.75 quantile gene expression. So again, in both cases, you can see that the imputed interval in orange, which basically just treats the predictions as gold standard labels, is an interval that completely misses on the true value, and that's because of prediction error. In our approach in green, the prediction powered intervals correct for prediction error and are valid, and they're also smaller than the classical intervals. Finally, we have several applications beyond just biology to other realms of science. So for example, here we're counting galaxies with computer vision models. So the labeled data set here is a small amount of data where images are labeled by expert astronomers as either being spiral galaxies or not spiral galaxies. In addition, we have a huge amount of unlabeled data that has classifications of spiral galaxy or not spiral galaxy based on a computer vision model. And the goal here is to estimate the frequency of spiral galaxies in the local universe. So once again, you can see the imputed interval, which just uses the predictions as gold standard labels, misses out on the ground truth. The prediction powered intervals correct for prediction error and are valid, while also being smaller than the classical intervals. All right, so as a high-level summary, prediction powered inference is a framework for using both your labeled gold standard data in addition to immense amounts of predictions to do provably valid statistical inference. You can do this with any machine learning model. So recall that I haven't made any assumptions about how you trained the predictive model F, its architecture, whatever. You can use whatever machine learning model that you've spent a lot of time on training and that you think is appropriate for the domain. We have this intuitive property that the better your predictions are, the smaller the resulting confidence intervals. And finally, the main technical meat of our work is proving that this procedure is statistically valid. So the intervals are guaranteed to contain the ground truth values with guaranteed high probability. All right, now moving forward, I'm really excited about applications. So I want to ask, what are the really big scientific questions that can only be answered with the unprecedented scale of information provided by machine learning predictions? So what big questions can we answer now that we have access to hundreds of millions of AlphaFold predicted structures, or hundreds of millions of predicted phenotypes, or predicted geographical origins, or protein functions? I want to be looking at, what are the kinds of questions that we can only answer with this new paradigm of doing science, and hopefully think about how prediction-powered inference can help make those questions more trustworthy to address. So this is something that I'd love to chat and brainstorm about. So if you have ideas or want to discuss this further, please feel free to reach out to me. And with that, thank you for your time. And thank you again to the organizers for inviting me to share this work. Huge thanks to all the people I collaborated with and received feedback on for this preprint. And I'm really happy to take any questions either now or here's my contact information if you'd like to chat further offline. Thanks.